Hello, and welcome to the fourth video in the Mitosis series, and this is actually the last one, so we're all done with that, which is great. And I just want to start with a review of telophase, because this is where we kind of left off, and that last picture was a little cheesy. So in telophase, which is, again, here, you know it's telophase because it's still only one cell, and you can see here that the centrioles are at the pole, spindle fibers are gone. The nuclear envelope is starting to come back. Now, these chromosomes have not really uncondensed yet. You can see it kind of better there. There's your nucleolus. So this here is cytokinesis. This is when the cells split. Now, you know that these are animal cells because they're round and there's no cell wall, but let's kind of review again real quick. So there's the whole story. Interphase, there's your chromatin. Once again, you can't see it. There's your nucleolus, no spindle fibers. Centrioles are at the poles. Prophase, metaphase, anaphase, telophase. Once again, this is not cytokinesis yet because these did not split. You know that as an animal cell, again, because it's round. There's no cell wall, and that is your cleavage furrow. We're going to pick up with that right now. So we talked about this as a review from yesterday. Cytokinesis is the division of the cytoplasm, and it happens, at the end of telophase. And what's going to form is a cell plate in plants and a cleavage furrow in animals. So let's take a little bit more of a closer look at that. This here is an animal cell once again. You know that because there's no cell wall. And the cleavage furrow is right there. It's kind of when the cell begins to pinch in. As it's pinching in, you're making a new membrane here. Once you make that new membrane, then the cell can split, and you're off to the races with two cells. 1, 2, and they're identical to each other. So that's an animal cell, cleavage furrow. And if you see here, these are some pretty zoomed in images, but you can see here how the cell's starting to pinch in right there, and you can see how it's starting to pinch in right there. So those are animal cells, those are zoom ins, and they are cleavage furrows. In a plant cell, it's called a cell plate, and you can see it's a plant cell because this cell wall, I don't know how much you remember from the old biology days, but that's your cell wall right there. And what happens is, since these guys do not end up just drifting off anywhere, they are still attached to each other because they're buds. You can see here how the cell plate is forming, and then it's forming more there, and then it's forming more here, until eventually you have a whole new cell wall. So that process takes time, obviously, but that is what is known as the cell plate formation, which will ultimately go on to build a new cell wall in plants. So that's your major difference with that. Quick review. One cell gives you two identical daughter cells. If all is good, chromosome number remains exactly the same. Chromosome structure remains exactly the same. No mistakes, it can go on to divide again, and that's the hope and the pray that happens in your cells. Quick little review once again. Plant cells do not have centrioles. They have centrosomes. Animal cells do. In a plant cell, we called it a cell plate. In an animal cell, we called it a cleavage furrow. So those are just kind of a little review box there. Now, let's control the cell cycle, because what if there's a mistake? If there's a mistake, the cell has 3 checkpoints that are very, very, very important. The first one is called the G1S checkpoint. So if we go ahead and we draw a little quick cell cycle here, there's mitosis, there's G1, there's S, there's G2. The first checkpoint happens right here. That's checkpoint 1. If the cell is not growing properly or it's made a mistake somewhere along the lines, the cell's like, nope, we ain't having it. Problem, abort. It's called apoptosis. But if it's growing fine and the organelles are great, perfect. We can move into DNA replication. Life is good, cell decides to divide. The next checkpoint is going to be right here. Now this checkpoint is going to say, okay, we've divided. Life is good. We have the right number of chromosomes. Everybody's happy. We can now enter into mitosis. So now we've committed, we're in. And the last checkpoint, I brought it up on the slide last time, is it's actually in mitosis. It's right there. It's in the middle of metaphase of mitosis. And that's where the cell has to decide, did the spindle fibers connect correctly? So you have checkpoint number 1, checkpoint number 2, and checkpoint number 3. If all of those check out, the cell divides and life is good. But could there be a problem? Well, there could be, so we'll talk about it. So late metaphase is your spindle checkpoint, your third checkpoint. So those 3 are important. Here's the same exact thing, but I just kind of shifted it around a little bit. There's that G1S checkpoint. There's that G2M checkpoint. And there's that one that's actually in mitosis. So those are your 3. Very important. So what's going to happen? Well, you're going to have these things called growth factors, and they're going to influence the cell cycle. So certain cells are triggered to grow at certain times in your life, or if there's certain kind of damages. And there's going to be a whole bunch of internal signaling, but again, we don't need that for this course. And that it can override, cellular controls that otherwise inhibit cell division. So let's see what we got. A couple of them are the platelet-derived growth factors, and this could, again, when you get hurt, this will trigger cells to divide to help you heal. And tissue repair is one of the reasons why we do mitosis. So if any of you have been cut over the last couple of days, well, you started to get better. That's good. Life is good with that. So this is an actual cell, and it's a plant cell, and you know that because there's squares. And this kind of has a little bit of everything going on in here. So let's see if we can see what's going on. This one is probably interphase, or it could be telophase. We don't know. Maybe it could be telophase because that one just finished too, so we don't know. One or the other. This one here is definitely anaphase, and you know that because you can see the chromosomes are moving apart. This one and this one are going to be metaphase. And this one here is probably prophase, and it's probably prophase because you can see the chromosomes are starting to condense. So that's kind of a real-life picture, and, you know, it's neat to see when it really happens like it's supposed to. And the final thing to discuss is cancer. In the event that your cell cycle controls go wrong, then your cells are going to replicate themselves out of control, and that is a major problem. So we got 2 things that could happen. You could have what is known as a tumor suppressor gene or a proto-oncogene. So we're going to talk pretty much about one only, but the 2 kinds of genes that can disturb the cycle when they're mutated. Now, everything's good and there's no mutation, then your cells are perfect and they fix themselves when they can and they go through apoptosis when they can't, and life is great. But if there's a mutation in any of those 2 genes, if there's a mutation there or there, then your cells could become cancerous. So a tumor suppressor gene, the best one that we've studied a lot recently, is this thing called the P53. And what will happen is, in the event that your cell realizes the DNA is messed up, there's damage to the DNA, the P53 gene is going to come in, halt cell division until it's fixed, and there's a whole series of events you do not need for this class, but it could happen, and life is great if it fixes. If it decides that, listen, there's nothing we can do, the cell is going to go through what is known as apoptosis, which is in essence cell death, programmed cell death, and life is good. The bad cell does not go on to damage the rest of your body. But if the P53 gene has a mutation, if it's not there, if it's damaged, whatever, now it doesn't know how to fix cells or realize there's a problem, and then these bad damaged cells go on to divide, and they form what is known as a cancer cell or a tumor. So our last picture, and mitosis is done. In the event that there's a problem, right there's your problem. P53 comes to the scene. It's either going to fix the DNA or do whatever it needs to do. Life is either great because you went on and you fixed it and life is awesome, or if you didn't fix it, you can destroy the bad cell and you're good to go, you're done. In the event that there is an abnormal problem, the P53 gene is messed up, well, maybe life will be okay and that's fine. But in the event that it takes the bottom path here, and there's no way to kind of destroy the bad cell, then you end up with this cancer cell. And what cancer cells, the reason why they're a problem is because they divide uncontrollably, especially where they don't belong. So if you end up getting a mass in your brain or in your breast or in your colon or anywhere, that cancer can grow and it could mess up the normal cells. And in some cases it's treatable and in some it's not. So mitosis is done. I got a bunch of practice things for you. There's going to be an internet project, and look for your test some point next week. Okay? So hopefully that was helpful, and I'll see you guys soon.